Hope they don't slip up. DNA. That's a good rap song. Because cancer is often caused by mutations when there's a problem with our DNA or there's mistakes at cell division. Our body slips up. So to understand cancer, we've got to understand cell division or mitosis. This is where the cells divide. They're going to replicate, is the word, their DNA. And we have to do that to grow or to repair our tissues. Now, in our body cells, all the chromosomes are diploid or 2N. That means there's a full set of chromosomes. And it all begins with interphase when the cell is preparing to divide. Okay, it's going to, now think about this. If it's going to divide, it has to replicate or duplicate everything so that when the division's over, the same number of chromosomes are present. Prophase, the second phase. I put P for prophase. All they're doing is the chromosomes are shortening into what are called chromatids. And at that time, the centrioles are just these little structures that hold the chromosomes together. They're going to move to the opposite poles of the cell. Finally, metaphase, chromosomes move together. It's like a barn dance. And it may seem like random, like who cares, but there's a reason to this, okay? Spindle fibers are going to attach to the centrioles and start to pull. And now we have the pulling of a part of the chromosomes called anaphase. And eventually telophase is when we call these daughter chromosomes, and they're identical in this process called cytokinesis, splitting of the cells. I remember this by, I polish my apples twice daily. Think of interphase, prophase, metaphase, telophase, daughter cells. I polish my apples twice. Okay, let's move on. Something a little more exciting. Meiosis. Meiosis is a reduction division, and that's where we have, instead of ending up with diploid cells, we have reduction down to haploid or N. And this ends up in a rearrangement, or we call it genetic recombination. And now our children are different than their parents. Now here's a nice little review. Let's just take a peek, lower right hand corner. I'm going to move it forward to where we kind of need to see. We don't care about that. There it is. We'll start with the cell. Now here's a review. Main phases: prophase, metaphase, anaphase, and telophase. And let's take a look. Okay, there's that cytokinesis occurring, splitting of the cells. And now the cells are going to prepare to divide. So we're going to go into probably interphase, I'm thinking. Let's see what they're going to name it. Yeah, interphase. Okay, it's preparing to divide. So we go from interphase to prophase, where, there we go. Now the chromosomes are becoming condensed, and we can actually see them. Okay, but under a microscope you can't even see these in interphase. But now in prophase we start to see them. They're pairing up. They're going to replicate, because remember, we got to divide this cell. So we have pairs of everything. Now at some point they're going to line up in that barn dance along the middle, and the centrioles are going to be attached by a spindle fiber. There we go. There's the spindle fiber on to metaphase. The attachment there. Now keep in mind, you know, this is not just some random process. This is how our cells divide. Every time we grow, like we started as a single cell, all of us, like we were one microscopic cell. And this process, like over and over and over, made us who we are. Even our, you know, our president, even movie stars. I mean, no matter who they are, they all started through this process of mitosis. Okay, come on, let's go on. Let's get on to, we're at metaphase. It's going on forever. Let's get to anaphase, where we're going to start to pull those guys apart. Come on, there it goes. And anaphase is going to lead to telophase. And then finally we get to split that cell. Okay, onwards, spermatogenesis. This is what occurs in the male seminiferous tubules. So they're the cells that are called spermatogonia. Now they're diploid. Now we can't, If we're going to make gametes like sperm, we can't have a diploid because we have to give half from the dad, half from the mom. So we need to divide that through spermatogenesis. Think of the Bible, Genesis, the beginning. There's those two end cells, and after meiosis, we end up with spermatids that are one end, these Sertoli cells, to basically nourish them and help them until they can swim. Oogenesis is when the primary follicle in the female ovary, we call that oocyte, and it's surrounded by some cells. Estrogen is going to trigger meiosis, and so now that primary follicle is going to divide into a secondary follicle, and eventually estrogen is going to trigger the formation of a grafting follicle. Now this one's, you can see it's swollen in the lower right, and during the menstrual cycle, ovulation, the ovum inside is going to be released. Now we can get on to cancer. We understand some basics of mitosis and chromosomes. Some characteristics of cancer, there's lots of them, many. One is they're non-specialized. They never become anything. Okay, they don't become skin, muscle, bone. They just divide and never specialize. And eventually they clog up parts of our system. Abnormal nuclei, they begin as regular cells, and that gradually that nucleus is going to enlarge. Here's a good example of a lower photograph. We start here with normal nuclei. Now they're getting larger, and finally in the far right, huge nuclei, and this is a cervical cancer. Defective chromosomes, due to, like, if there's too many copies, and that's due to mistakes at mitosis, or there's deletions, whatever the process, usually our body undergoes apoptosis, and that means we kind of have a programmed cell death. We destroy cancers every day of our life. But areas with frequent cell division, I'm thinking of like the lining of the lungs, the GI tract, the skin, anywhere where there's rapid mitosis, these are also the areas likely to become cancerous. The telomeres are the kind of the end caps. If you look at the lower left, you see a chromosome, and the telomeres at the tip. And normally, when after a certain number of cell divisions, there's the telomere is going to do this programmed death where it binds the ends together, and we have apoptosis and death. But not so with cancers. They have this telomerase enzyme that preserves the telomeres. So these cancer chromosomes are basically immortal. They just keep dividing and dividing, and they never fold on themselves. And that's a problem. Carcinogenesis, think of genesis again, the beginning of cancer. Most normal cells have this contact inhibition. So when they're growing and they run into something, let's say they run into the blood vessel, they stop when they contact a neighboring tissue. But cancer cells don't care. They don't care if there's bone in the way. They don't care if there's muscle tissue. They're going to continue dividing, forming these large tumors, and eventually spread through a process called metastasis. Now, malignant tumor, that refers to a cancer that is no longer in situ. It's going to spread. Either they expand locally just by growing and spreading, or we have this process called metastasis, in which a cancer cell spreads from a core neoplasm. And it's going to use the lymphatic or circular system as a vehicle to colonize new sites. And there are distinct stages to these metastasis. Initiation, that's an early stage where it's often undetected. There's no symptoms. There's just a mutated gene that has not been programmed to die. Then promotion, the tumor is going to develop and we get a mass. Still no symptoms usually. Progression, now we have the invasion of the tumor into underlying tissues, but we don't have metastasis yet. It's not on the move. It's still in situ. Malignant tumor, now we have a problem because it's invaded the lymphatic, which is indicated with a green blood vessel, and blood vessels on the red. Distant tumors, now we have these satellites that are, so you might start with a lung cancer, and it's going to move into the bone and into the prostate or wherever it wants to go. It's on the move. Metastasis. Angiogenesis, again, genesis, the beginning. Angio, think of blood vessels. So if you're a cancer tumor and you're on the inside of that mass, you're getting almost no oxygen and almost no glucose. And you're going to starve unless somehow your tumor can release these angiogenic growth factors. And that's going to stimulate the development of blood vessels that are going to surround and support the tumor. So the tumor is kind of tricking the body into feeding it. And I guess it makes sense because the tumor is us, right? It's not some evil thing. Cancer is us. And this angiogenesis is one way that we have learned to starve tumors by cutting off their blood supply. Some of the causes of cancer, well, heredity, that's huge. There's still debate about this, but all of our genes have what are called tumor-suppressor genes, thankfully. And this often occurs at night while we're sleeping. These tumor-suppressor genes have an enzyme that goes along and checks our DNA and makes sure it's not cancerous. And that's another good reason to sleep at night, okay? At least 6 hours. If we have a mutation of a suppressor gene, such as the BRCA, those are genes that suppress breast cancer. But if there's a mutation there, and this can be tested, then a woman is at risk of developing breast cancer. The radiation, okay, we can't always avoid radiation, but we can put a hat on out in the sun, and we can limit some of our exposure to things like radon, nuclear fuels, of course. X-rays, to be honest, there's a very brief blast of radiation. It's really not very dangerous. Most of our radiology has really pretty advanced and safe. Skin cancer, melanoma, frighteningly, 2% of the total cancer deaths each year due to just this one type of skin cancer. It has a brown, irregular, raised margin, and it's not always on skin surfaces exposed to sun. Sometimes it's in the auditory canal of the ear, or between the toes, or perianal. So at some point, it's good to get a full body examination. Viruses. There are viruses that are sneaking away. They can insert their DNA into their host. They're called lysogenic viruses. And hepatitis B is known for that. It starts off as a hep B infection, and it'll move into the liver and as HPV. And this is one that we can prevent with a vaccine. Inhaled organic chemicals, we think of industrial solvents, tobacco smoke, anything that can trigger these cancer-susceptible genes that we have. And speaking of cancer, let's look at this. This is a nice little review of this lecture. Okay, we start with a cell. There is simple mitosis. Remember, mitosis produces identical cells, interphase, prophase, metaphase, telophase, daughter cells. Meiosis is different. We're going to have, that's not meiosis. That is cancer. Because you can see the large nucleus. And remember, cancer cells don't become anything. They develop, and it's becoming, there we go. It's invading, so it's invading local tissues. And at some point, that cancer is going to metastasize. And metastasis is when it's no longer in situ, but is going to use the vascular system. So there's a lymphatic. It's invading the lymph system, moving who knows where, into bone, maybe muscle. Oh, and now it's going into the circulatory system. Well, do your best out there. Try to live a clean life, eat right, and get screenings. All right. Thanks for listening.